Hello, and thank you for joining our webinar today. We'll be discussing how single-cell multiomics can uniquely inform on clinical precision oncology and how Curiox's Laminar Wash next-generation sample prep technology supports these efforts. I'm your host, Dr. Jeffrey Feld. Our speakers today are Dr. Jonathan Scolnick, founder and chief science officer of Proteona, a precision oncology company based in Singapore, and Dr. Chyan Ying Ke, Associate Director of Bioapplications at Curiox Biosystems, and she is also based in Singapore. Jonathan completed his Ph.D. at UC Berkeley and postdoctoral work at the Salk Institute in La Jolla, California, before joining the molecular engineering lab of Dr. Cindy Brenner at the A-STAR in 2009. Following his time at A-STAR, Jonathan returned to the US, where he developed multiple genetic diagnostic tests at Natera, as well as a line of targeted sequencing products for NuGen Technologies. In 2015, he returned to Singapore, joining the Department of Physiology at the National University to continue developing new assays in the single-cell space. In 2017, he co-founded Proteona, precision medicine, to help bring single-cell biology into the clinic. Chyan Ying obtained her Ph.D. in mechanical engineering from Harvard-MIT Division of Health Sciences and Technology in Massachusetts, where she studied the modulation of B-cell adaptive immune response with nanoscale organization of antigen presentation on biomaterials. She furthered her interest in integrating biocompatible materials in biological applications at the Singapore Immunology Network A-STAR in Singapore. Her pursuits in bioengineering, immunology, and innovation led her to her current role as Associate Director of Bioapplications at Curiox, where she leads the development of enabling research and relevant applications using the Laminar Wash technology while participating in product design and development. After the webinar, registrants will receive a link to access on demand. Let's go ahead and get started with Chyan Ying, who will introduce Laminar Wash and its utility for single-cell omics. Jonathan will then tell us about how Proteona approaches single-cell omics to inform cancer therapy, and Chyan Ying will give us a summary. We should have plenty of time for questions, so please use the question interface on your screen. We're also trying out a new function this webinar, this poll question. Most folks have actually responded, but if you haven't already, we'd really appreciate if you can kindly answer the poll question on whether you use multiomics and cell hashing in your experiments. And it looks like we actually have a lot of folks who don't use single-cell genomics, so hopefully this should be a really informative webinar for you. So without further ado, I'd love to introduce Dr. Chyan Ying Ke.
Hello, thank you, Jeff, for the kind introduction. Welcome to Curiox's joint webinar with Proteona today. So I'll first give an introduction on Curiox's Laminar Wash technology and its applications in your single-cell assays. Following that, Dr. Jonathan Scolnick will share about Proteona's approach to single-cell multiomics and the insights they have for precision oncology. Jonathan will further elaborate on workflow challenges associated with single-cell multiomics and how Proteona has adopted Laminar Wash in their workflow to address these challenges in preparing clinical samples in their workflow. So cellular analytical tools have come a long way, and through modern techniques like flow and mass cytometry, as well as single-cell multiomics, we can now probe phenotype, functional, and transcriptomic information all from the same cell. These advances have accelerated applications in vaccine development, oncology treatments, organ transplant, just to name a few. Yet, you'll be surprised to know that the key component of sample preparation, which is centrifugation, remained largely unchanged since the 19th century. Centrifugation is a process that is difficult to automate and lends itself to inter- and intra-user variability, challenging consistency and reproducibility in your assays. We will also look at some data in this webinar on the harsh impact of centrifugation on your precious cell samples. So it's about time for sample preparation technology to catch up. Laminar Wash addresses the problems with centrifuge during sample preparation, offering an automated washing process that is fast, reproducible, while being gentle on cells. Well, to begin, many researchers have taken the centrifugation step for granted, as until recently, it has been the only method to remove excess reagents from suspension cells. But in this image, showing cells expressing endogenous red fluorescent protein, or RFP, spiked in with non-fluorescing cells, samples washed by centrifuge are observed to be lower in count, distorted in morphology, and importantly, have lower expression of RFP. This can be contrasted with the cells that were prepared by laminar wash. The cells appear viable and are significantly brighter in red fluorescence. Now, if you look at the big picture, the red fluorescent protein in this image can represent any protein of interest in your cells, be it chimeric antigen receptor or CAR, checkpoint blockade proteins, tumor antigen, or even that elusive biomarker that is sensitive to mechanical handling. It is now possible to wash your cells to reduce non-specific background staining while maintaining fidelity to protein expression using laminar wash. And the problem of non-uniform cell loss in centrifugation is also a well-known issue. This leukocyte staining from whole blood showed that neutrophils and myeloid cells are poorly retained or stained in the centrifuge sample, but the same populations are prominent in the laminar wash prepared samples. This result has been reproduced in other flow and mass cytometry experiments, showing loss of cells specifically in the myeloid compartment when centrifuge is the method of preparation. So let's talk about how laminar wash is able to retain these cells so well. Curiox's laminar wash is characterized by a unique plate design where we have hydrophilic wells surrounded by an ultra-hydrophobic surface, forming a virtual wall. The hydrophilic wells accommodates the buffer and cells, while the hydrophobic surface enables droplet formation in the wells without cross-contamination. The wells can accommodate 70 microliters of fluid and up to 150 microliters with the use of a large volume adapter. During incubation with reagents or antibodies on laminar wash plate, the cells naturally settle to the surface of the well by gravity. Unlike pelleting by centrifugation, this ensures that cell physiology remain undisturbed. The unbound reagents, cellular debris, and less dense particles remain in the bulk buffer solution. And after cells have settled on the plate can be loaded on the washer platform. You can customize the buffer flow rate and number of washers, accomplishing the equivalent of 2 rounds of centrifuge in as little as 3 minutes. In the washer, 2 nozzles are lowered to each well. One nozzle to dispense fresh buffer, and another nozzle to aspirate waste. The debris and particles in suspension are aspirated, while the settled cells are undisturbed due to the low flow rate of buffer exchange. Through multiple dispense and aspiration cycles, laminar wash can accomplish zero dilution in a short amount of time, while the cells remain undisturbed and uncrushed by centrifugation forces. The laminar wash family of systems has a variety of throughput and automation level to suit every lab's workflow and budget. The compact-sized Mini has an 8-well washing throughput from a 16-well strip and is popular among mass cytometry and single-cell sequencing users. For higher throughput and streamlined workflow, the HD2000, shown here with an LCD touchscreen interface and an optional automated buffer exchanger, processes 96 wells at once. This is the ideal platform for cell hashing of multiple cell samples in preparation for site-seq workflow. The Auto 1000 integrates the HD2000 and buffer exchanger in a Hamilton Nimbus automated liquid handling system. The platform is designed to produce the most quantitative and reproducible results for flow cytometry users by reducing user variability and day-to-day variation prevalent in manual sample handling. And here, I would like to quickly illustrate how simple and intuitive it is to transit from conventional centrifugation workflow to laminar wash, using the HD2000 and a simple staining protocol as an example. The HD2000 is extremely compact and can be easily set up in the biosafety cabinet. We first prepare samples in the laminar wash 96-well plate, similarly to how you would prepare in a microtiter plate. Cells can be added directly onto the plate, followed by addition of blocking buffers or staining reagents. During incubation, cells naturally settle to the bottom of the wells by gravity. Prior to washing the cells, place the plate on the stage and choose the number of wash cycles and buffer flow rate, which can be easily customized using the touch screen and adapted to your protocol. Pressing start retracts the stage and initiates the wash. So you're now free to grab a coffee, since a 9-minute wash takes only about 2 minutes. For site-seq protocols, we recommend a 25-cycle wash, which takes about 6 minutes. At the end of the wash, Every well has a uniform volume of 25 microliters. The cells are now ready for collection to count and for the next step in sequencing. In the next couple of slides, I just want to quickly highlight some data we have run with collaborators, and it will complement the data that Jonathan will present. The following examples show optimized laminar wash methods. We work with each customer to optimize the specific protocols. So first, we'd like to highlight a recent publication that validated the strengths of laminar wash in single-cell sequencing. The group at University of Surrey in UK wanted to connect single-cell transcriptomics level resolution of peripheral B cells to their well-characterized phenotypes. Because B cells are a low-frequency population, many single-cell RNA studies tend to group different phenotypes together, possibly missing on important disease-related distinctions and insights. So the group started with their primary sample, which was PBMCs from a healthy male, and sorted them into 5 well-characterized phenotypes by FACS, distinguished by CD19, CD27, and IgD expression. The sorted cells were then sequenced individually on chromium 10x on 5 lanes. With 2 separate validation samples, they performed a similar sort to obtain 5 phenotypes, then hatched them with BioLegend's Total C kit, using Laminar Wash Mini 1000 washer to perform the cell washing. The total of 10 samples were then combined to acquire with chromium 10x on a single lane. Comparing the UMAP analysis for primary donors and the 2 validation donors, the colored boxes and circles align the phenotypic identities with corresponding groups on the transcriptomic map. The authors note that the validation maps recapitulate the primary donor map, despite using only 1 lane on 10x instead of 10, if the phenotypes had been acquired separately. Overall, Laminar Wash process samples recapitulated the primary map despite using only 1/10 the number of cells analyzed per donor. Instead of spending resources to run the 5 B-cell phenotypes in 2 validation samples using 10 lanes, the authors used total cell hashing to acquire all 10 samples together on 1 lane. Laminar Wash ensures success, as noted by the lead author, Dr. Alex Stewart, who commented that there is less background after staining and washing on Laminar Wash, enabling clear differentiation of samples. Alex was also excited that the team did not have to spend as much time washing cells, as the Mini did all the washes in a one-touch walk-away step. Assays with isolated nuclei are particularly challenging, as nuclei are physically smaller than cells, making the nuclei palette difficult to visualize after centrifuge wash. As nuclei have a larger mass density than cells, they settle on laminar wash plates swiftly and can be washed gently without clumping or nuclear loss caused by centrifuge. Cerivance is a biotech company interested in developing therapeutics for central nervous system diseases. They evaluated the relative retention of nuclei on Laminar Wash Mini, and they could obtain They evaluated the relative retention of nuclei on Laminar Wash Mini versus retention on a centrifuge. At all of the starting nuclei numbers, Laminar Wash showed greater retention of nuclei compared to centrifuge. More importantly, Cerivance showed that even with 10-fold less antibodies used for staining on Laminar Wash, they could obtain the same staining resolution as centrifuge that used the regular titer of antibodies, suggesting that users can save on expensive oligoantibodies while obtaining similar phenotyping results with Laminar Wash. Here at Curiox, we isolated nuclei in bulk and proceeded to wash by either centrifuge or Laminar Wash. The nuclei are then stained by trypan blue and enumerated with cell counter. In the duplicate wells washed by centrifuge, large clumps can be observed in one sample, while the second well experienced large loss of nuclei. This is in contrast to Laminar Wash that have consistent nuclear retention while remaining distinctly separated. The automation of washing by Laminar Wash also serves to reduce variability between operators. Here we have an example showing mouse tumor infiltrating lymphocyte with immunophenotyping on flow cytometry. The heterogeneity and necrotic nature of tumor samples lends to a preparation of high debris and non-viable cells. And moreover, the complexity of working with tumor models can be demonstrated further by centrifuge samples prepared by different operators versus Laminar Wash. Summing up here, the analysis of tumor cell suspensions produces varying degrees of debris, and Laminar Wash effectively reduces that. The results seen here from manual operator 1 and 2, which employ centrifuge washing, represents different operator experiences and seniority when it comes to sample processing. The efficiency of Laminar Wash is not limited even at higher cell densities, ranging from 1 million, 2 million, and even up to 5 million tumor cells. In this comparison that you see, the reduction of debris was seen to have improved from 4% down to 2% in Laminar Wash, regardless of the experience of the operator. The live cell staining results here indicate a better resolution achieved when the tumor cells are non-pelleted. The viability of TILs have been improved from 67 to 88%, enabling greater confidence in immunoprofiling of these tumor-infiltrating lymphocytes. The analysis here can be extrapolated to sample preparation for single-cell sequencing, where you want to ensure you maximize the number of viable and intact cells that make it to the sequencing module reproducibly. So with that, I'll hand it over to Jonathan, who will elaborate on Proteona's experiences with adopting Laminar Wash in their preparation of clinical samples for multiomics analysis. Jonathan, please.
Great, thank you, Chyan Ying. So at Proteona, we were really focused on utilizing single-cell multiomics tools to improve patient outcomes. And we do that through a combination of our proprietary wet lab and data analysis tools. So the reason we focus on single-cell biology is because we know that tumors are very heterogeneous. Not only do they contain a number of different cell types, as shown on the left, but even within a tumor itself, the cancer cells themselves are heterogeneous, and that heterogeneity can lead to therapy resistance. But it also, understanding that heterogeneity can give us a hint as to how to improve care for a given patient. And so because of that, we've developed a set of tools to create a biomarker discovery pipeline. And our focus is really on understanding how to predict drug response to improve the therapy that a patient is going to be getting. And as I mentioned, we have both wet lab and dry lab tools to do that. On the wet lab side, we use what we call our ESCAPE platform, which is a platform developed for simultaneous detection of both protein and gene expression. We use the 10x genomics system to create our single-cell sequencing libraries. And as part of that, we can also add to it TCR, BCR sequencing, as well as targeted mutation analysis, depending on the particular problem that we're looking at. And of course, we spend really a lot of our time, most of our time now, on data analysis. And so if we dig a little deeper into how we think about data analysis, we sort of break it down into 4 parts. And for each of those parts, we have a particular tool that we like to use. The first thing that we would do with a data set is use a tool we call MapCell, and this is used for annotating samples, so identifying the cell types that are present. Then we use a tool called MapBatch for removing batch effects between samples. MapResearch is really our tool for biomarker discovery. And then when we use a tool called MapResponse, which is a tool that then helps predict how is the next patient going to respond to a particular therapy. And I'll go into a bit more detail on each of those in subsequent slides. But I'll start with an overview of our wet lab procedures, because this is why I was invited to give a talk today. And so we start with cells, and as I mentioned, we tend to focus on tumors, and so these can be anything ranging from bone marrow aspirates to fine needle aspirates of solid tumors. And so we often have little to no control really over how many cells we're receiving and kind of how good the cells are, how viable they are. So we receive these cells and we stain them with our antibody pools. And this is just like a standard flow cytometry experiment setup, but instead of using fluorescent antibodies like we would use for flow cytometry, we use these DNA barcoded antibodies. so that subsequently the readout of antibody binding is based upon high throughput sequencing analysis. Now, for all of our samples, we go from antibody staining directly into the Curiox cell washer. We like the laminar flow system because it provides a very standardized washing setup, really reduces the hands-on time, which is a big bonus for us. And then on the right side, we have this graph that also shows the cell retention that Chyan Ying mentioned from another group. And it really allows us to take these small samples from tumors and be able to still stain them and wash them and retain enough cells to do our sequencing preps. So why do we make measurements of both protein and gene expression? It's easy to think that those might always be the same, and so you would only need one modality, but it turns out they can be very different. And by looking at both protein and gene expression, you can add a lot of additional information to your samples. So in this experiment to show that, we've taken normal human PBMCs and we activated them with PMA and ionomycin and we either had untreated or treated them for 1 hour or a 16-hour time point. And then we looked subsequently at the gene and protein expression and wanted to see how frequently the patterns were the same versus how often they were different. And so if we look at just two sort of standard markers, we can look at CD3 protein expression. And so we can see that in untreated samples, we have a baseline amount of CD3 that decreases at the 1 hour time point because we're measuring surface CD3. And when the T cells are activated, they actually internalize the T cell receptor complex. And then over time, at the 16-hour time point, we see a rebound or back to normal expression levels on the surface. And at the RNA side, we see basically the same pattern. So some baseline amount, a decrease at the 1 hour time point, and an increase again at the 16-hour time point. So here you would say that the CD3 protein and the RNA signals are a good match for each other. But if you look instead at CD69, which is a marker of cell activation, we can see a baseline amount of CD69 protein expression that increases quite a lot at the 1 hour time point, like you would expect, and then starts to decrease a little bit at the 16-hour time point as the cells attenuate to the presence of the PMA and ionomycin. But if you look at the RNA level, you see something very different. So you have a baseline amount of gene expression that starts to increase at the 1 hour time point, but then continues to increase at the 16-hour time point. So you can imagine now that if you were looking at only protein or only RNA, you would be getting two very different answers about the state that these cells are in. And in this particular experiment, when we looked across these activated PBMCs over time, we saw that 25% of the proteins measured showed a differential expression pattern compared to the RNA. So as I said, we really do feel that adding the protein information to the gene expression really adds a lot of additional depth to the data that we're collecting. So for the rest of the talk, I'm going to focus in on an example application, which is a project we've done looking at multiple myeloma. So multiple myeloma is a cancer of the plasma cells in the bone marrow. So these are the antibody-secreting cells. And the really important feature for us about multiple myeloma is that it's currently not curable. So patients can survive for quite a long time, but following the initial relapse, patients tend to relapse every 12 to 18 months. So roughly every 12 to 18 months, a new drug treatment has to be given. Patients, if the patient responds to that, then they get about another 12 to 18 months before they have to move on to their next treatment. So it's a difficult disease, both to live with and to treat. Now, this particular study was quite small, so 17 patients. We had samples from both pre and post treatment. And our goal was to really just show how Proteona's tools could be used to study disease. And sort of the stretch goal was to see if even with this limited number of patient samples, we could start to identify biomarkers that would correlate with how the patients respond to therapy. So as I mentioned before, the first thing we do is annotate the different cell types across the samples. using MapCell. And what I just want to point out here is this purple box, which we call novel, which means that this cell type or cell state is novel to our algorithm MapCell. And in MapCell, we allow for this novelty because most algorithms will take a sort of cell type that maybe the algorithm doesn't know very well, and it will overfit it into the nearest kind of cell type. And instead, we want to make sure that we're always identifying what the cell types or cell states actually are. And so if our MapCell algorithm doesn't know the answer very confidently, it will basically call it novel, raise a flag, and then allows us to go in and investigate further. The next step would be to do batch correction with our tool MapBatch. So if you look on the left-hand side, we have a UMAP plot. So each dot here represents data from a single cell. And then the cells here are colored based upon the sample that they were derived from. And you can see basically at the raw data level, it's a bit of a mess. And the cells are clustering themselves based upon which sample they came from rather than which cell type they are. So that's not very helpful for downstream analysis. But once we do the batch correction, we can see that the cells now overlap, and instead of clustering by sample ID, they cluster by cell type. And now with batch correction, there's also similar to cell annotation, there's a major problem that you have to look out for, which is that most batch correction algorithms will actually overcorrect and sort of force you to lose biologically relevant information because cell types happen to be similar enough to each other that the batch correction algorithm decided that they needed to be put together into one cluster. And so MapBatch, again, tries to avoid doing that and will leave a little bit of sort of batch effect in place in order to make sure that we don't lose the biologically meaningful information. And in this study, in the red circle here, we were actually able to identify 4 new cell states. So that is cells where MapCell had not seen them before. So they were identified as novel or unknown. But they also formed their own individual clusters in this general area. So subsequently, we've been able to take those cell states and use unrelated data sets, both internally and from the literature, and we can identify these 4 cell states as independent cells, or rather independent cell types or states. And as I alluded to on the previous slide, we also have taken this data set and run it through standard published batch correction methods. And we find that in all of them, or rather in none of them, can we find all 4 of these cell states. But some of them are a little better than others at finding 1 or 2. And again, really, one of the worst things to me that you can do is lose important data because you overcorrected it. Now, that overcorrection in this case would actually be really harmful because it turns out that these cell types or cell states actually have a strong correlation with how the patients progressed in their disease. So we were able to take those cell states and identify a gene signature that correlated quite well and significantly with progression-free survival. So patients who had low gene signature tended to progress or tended to not progress rather over a long period of time, whereas patients who had a higher level of this gene signature, meaning more cells expressing that gene signature, did not perform as well during therapy. And one of the really interesting pieces of information here is that this cell signature is found in fewer than 1% of the total cells collected. So this is not something that we would be able to identify from any type of bulk analysis. It could only be identified through single cell types of analysis. And really here, we could only identify it because we didn't overcorrect our samples, and we had both the batch processing not overcorrecting, but we also had MapCell telling us that these were cells that we should look into. And so this is a nice correlation between this gene signature and progression-free survival. But we want to always go to the next step, which is this map response tool. and say, okay, can I now use information that we've found previously to predict the outcome of the next patient? And for this, we rely heavily on machine learning. So we basically learn the response of a patient and learn the features of our data that correlate with that response in order to develop classifiers so that we can classify the future patients into how we think they will respond to the treatment. And here, this is a bit busy of a slide, but we have our 17 patient samples, and for each one of them, we're testing the accuracy of 4 different classifiers. Each of those classifiers use slightly different aspects of the data. And we could see that even with this very small data set of only 17 patient samples, we could generate a classifier that could predict up to 88% accuracy how the patient was going to respond to a particular treatment. Now, just to sum up, again, Proteona as a company is really focused on clinical applications of single-cell biology, in particular focusing on how to predict a patient response to a given therapy. And that can be across many different tumor types, as well as cell therapies. We do that with both wet lab and dry lab analysis tools. I showed you that we were able to build a gene signature classifier that corresponded to treatment response, even though that classifier was only present in less than 1% of the total cells collected. We were also able to build a predictive treatment response classifier with up to 88% accuracy. And wanted to point out, again, just that we use the Curiox mini cell washer with all of our samples because it really standardizes the workflows and improves our throughput. So I'm going to stop there. Here's an email address you can send a message to if you have any questions. We're going to go back to Chyan Ying now, and I'll stick around for questions afterwards. Thank you.
All right. Thank you, Jonathan, for sharing the exciting work that Proteona is doing. I think we are all keen to experience the improvement of data quality made possible by Laminar Wash. So to round out this seminar, I would like to highlight that Laminar Wash is adopted by major biopharma companies in a preparation workflow for single-cell sequencing. And BioLegend recommends the use of Curiox Laminar Wash Mini in their total-seq protocol for high cell recovery and debris removal. We just had an exciting joint webinar with BioLegend and Immunai, with both sharing their data from optimizing total-seq assays on Laminar Wash. So do check that out on our website as well. In summary, Laminar Wash technology streamlines the sample preparation process through workflow improvement and producing higher quality data. With Laminar Wash, users can now perform the cell washing with less hands-on time while achieving consistency and reproducibility independent of operator and location. Semi and full automation options are available to customize to the needs of your lab. In addition, Laminar Wash washers are compact enough to fit in biosafety cabinet, limiting aerosol exposure to the operators. Cells prepared by Laminar Wash are closer to their native state with increased retention and viability. Debris removal is enhanced, ensuring that you are sequencing precious cells and not junk. In addition, the enhanced washing efficiency over centrifuge provides clear distinction of cell hashing and cellular phenotype, improving your multiomics data. So with that, we thank you for your interest and attention, and Jonathan and I will be happy to take any questions now.
Thank you very much to our speakers, Chyan Ying and Jonathan. Very exciting talk. I'm happy to take any questions. There's been a couple that have popped through both before the webinar began. Folks were able to enter questions as they registered, and we've had a couple of questions pop up here. So this first one, I believe, is probably best answered by Chyan Ying. Can we use the washed Laminar Wash plate with now washed cells? Can we use those directly on a flow cytometer?
Yes, you can, absolutely. So our 96 well plate is SBS formatted, and that will fit perfectly with any flow cytometer. We may need a couple of accessories to ensure the high volume on the plate for proper acquisition. But I think you can talk to your local FAS regarding that, and we'll be happy to guide you through direct acquisition from the plate.
Thank you. I think another question for Chyan Ying. How many cells do you load onto one well on the laminar wash plate?
So yeah, that's a great question, and I think some of this really depends also on the sample type you're using. So in Curiox R&D itself, we have looked at up to 10 million PBMCs on each well of the laminar wash plate, and have shown that even with 10 million PBMCs, there is similar retention across different starting cell numbers, as well as a similar staining index, showing that the wash efficiency is very comparable regardless of number of cells on the plate.
Great. So very variable. Really, how many cells you have, the instrument can probably wash them. For Jonathan, so how many samples do you typically run at a time when you have samples coming in? Is it one? Is it more?
Yeah, so it really depends on the particular project. So if we have a small needle biopsy, that's something that has to be processed immediately, and we get a call from the hospital saying a sample's coming in, and we'll process it right then. If cells can be frozen or cryopreserved, we tend to then expand the number of samples we look at a time, and we tend to batch in groups of 8 because that fits well on both the cell washer as well as other sort of handling platforms we deal with.
Great. Yeah. So you can basically handle any number of samples depending on their state and how they're coming in. Good. We got some more questions coming in. So just kind of a two-part question. How efficient is the laminar washing? And then second part, laminar wash, can it be used for any kind of cell?
Okay, I'll take that question. So I think the neat thing about laminar wash is unlike centrifugation, on the laminar wash platform, we are really doing sequential dilution of the reagents with each addition and aspiration of fresh wash buffer. And so, we estimate that in a 9 times wash cycle on laminar wash, we should in theory hit up to 17,000 times dilution of your original reagents. And if you just contrast that with what you do on a centrifuge, which typically if you're using a tube, you would dilute 100 microliters of reagent with 1 ml of buffer, that would be a 10-time dilution. If you do a twice centrifuge, that's anywhere from 100 to 1,000 times dilution. So it really goes to show that on the laminar wash, you can achieve significantly greater wash efficiency in less time, since a 9x wash takes about 3 minutes. And then the second question is, what kind of samples can we wash on laminar wash, right? And that is a great question, because the cool thing is that on laminar wash, you're really depending on gravity to settle the cells. And so any sample type that is denser than the buffer that you're washing with or incubating with will settle and will wash efficiently. So we have, as I mentioned earlier, showed data on nuclei and cells, an obvious example. We have also successfully done even smaller organisms like yeast and bacteria. We've done, multiple other cell types, brain cells. Some of them could be quite challenging because of the amount of debris, amount of fatty tissues that they have. But I think in most of them, we have had a successful demonstration of retention.
Thank you. And along those lines, in terms of the efficiency, since you can control the flow rate, you can control the number of washes. Obviously, you can you can adopt your current SOP, your standard of practice methods to laminar wash, but it also gives the opportunity to really optimize your wash. If you have a lot of background, you can remove it. If you have, depending on your experiment, you can really customize. So this question has to do with cells settling down by gravity. So since cells are denser than the buffer, how do you decide which cells are denser? And for that purpose, do we need to do any kind of standardization? So really just thinking about my cells and how are they going to settle? What settling times do I need to consider? Is it cell specific?
Yeah. So I think thankfully most cells I think would be pretty similar in terms of their overall mass density, and of course the cell type that we have worked with most commonly and is most commonly used by our users would be PBMCs, which are actually relatively small in physical size, and with that, if just to give PBMCs as an example, if you're starting with 1 million PBMCs, it takes about anywhere from 20 minutes to 30 minutes of settling time, during which this time is also used for the incubation together with the antibodies. So there really isn't any, there really is hardly any increment of time to your workflow. And as for other cell types, I think for most of them, the mass density will hover around the same values, and I think we can always, we could always start from the ballpark time with PBMCs, and then kind of optimize based on your retention requirements as well as your workflow requirements on the exact settling time for your protocol.
Thank you. So how would you define an unsuccessful wash? What criteria do we typically think about if it's a wash is unsuccessful?
I think Jonathan might be a good person to get that.
Sure, yeah. So this is a little bit of a trick with the Curiox system in that if there's an unsuccessful wash, you can't 100% of the time figure that out before you go do your downstream analysis. So in the case of flow cytometry, what you would see is just a lot of cells being stained, like high sort of high non-specific staining. In the sequencing side, you basically get the same phenotype. It just takes you a few more days to figure that out. Internally, we've developed some SOPs now to be able to track and make sure that we don't have any unexpected sample clogs or something like that.
So really just typically the downstream analysis is going to tell you whether you had an unsuccessful wash, and it may just be the overall sample prep itself. The centrifuge, if you had gone more of a classical route, may give you the same problem if it's an unextensive wash, or if settling wasn't long enough, or too long or too short of an incubation time. Here's a really good question. How is cell clumping avoided?
Jonathan, maybe you want to take that first?
Oh, okay. Sure. I mean, from my perspective, the cells don't clump because they're never forced together. So they settle onto the bottom of the plate, just by gravity settling, and then solution just flows basically on top of them. So they're not pushed around, they're not shoved into one side of the well. So there's no force acting on the cells to bring them together. I think that's why they don't clump, compared to centrifugation, where literally that's the purpose of what you're doing, is to force all the cells to into a small space.
Right, thanks. So I think maybe just a little bit, maybe I'll just elaborate a little bit more on the application area. I think for a lot of common sample types that we work with, PBMCs, as Jonathan mentioned, since they don't undergo all that mechanical stress, we don't really see a huge amount of clumping with those. And of course, there are more challenging sample types like a tumor cells. As we have elaborated earlier, they are necrotic. They come from a very disorganized network, and I think a lot of some of these clumping definitely will be avoided using laminar wash, but some additional tricks like perhaps having enzymatic digestion, the use of DNAs would also help to improve the quality of the samples as a whole, regardless whether you're using laminar wash or centrifuge.
Thank you. Yeah, so typically you don't have to worry so much about cells clumping since you're not forcing them together, but there are some tricks that are pretty common out there to try to declump your cells if you do end up with a difficult sample type. And go ahead and follow up on what Chyan Ying just mentioned too. If you're working with difficult tumor-derived samples, like you're looking at tumor infiltrating lymphocytes, or some of those real difficult, notoriously difficult samples to work with, definitely encourage folks to check out our most recent webinar before this one back in May, the flow cytometry UK webinar we did with Charles River. We talked a little bit about TILs and working with those types of samples. So here's a follow-up question to cell density. If you have a cell whose nature of the cell is actually to float, is that going to be a property that might be of concern for laminar wash?
I think you have absolutely gotten the gist of the principles of laminar wash. So I think you're right. I think so far we have not yet encountered adipose cells, fatty cells, but our guess is that those are not going to work really well on the laminar wash. And even cell types that are commonly associated with neighboring fatty tissues, for example, brain cells. I mean, for those, if you are looking at, for example, brain-associated leukocytes, we also strongly encourage the pre-purification of the leukocytes from the surrounding, very high fat content cells in order to ensure better settling on the laminar wash platform.
And on the flip side of that too, dead cells tend to float. So if you have a sample, with difficult to work with cells that are dead or dying upon handling, you're going to wash those away and not have to worry about counting those. I had a question about just with the batch correction, Jonathan, and maybe since you said that you use the laminar wash systems for all your samples, so I was just curious if you noticed when you first adopted laminar wash, was there any improvement in the batch correction since you're seeing, working with more standardized sample preparation, kind of taking some of the inter-operator variability out? Was there any impact, do you remember, on batch correction?
Yeah, that's a great question. I don't have a good answer for you. We've been using the mini system since it was in beta. And so it's been so long now, I don't really remember what life was like beforehand. So I don't have a good specific answer to that question. But we do know that most of the batchiness of our samples comes from the way the samples were handled before they got to us. So, we get samples collected from different clinical sites, literally different countries. Each of them can have different handling procedures. And so that's generally what we see as the biggest drivers of batch effect.
Gotcha. That makes a lot of sense. Definitely the site collection, depending on time of day and how they were handled, that can have a huge impact on something so sensitive like single cell omics. Great. Well, this has been very informative. Thank you so much to our speakers. We should have another webinar coming up in the next couple of months, so stay tuned. We also will be sending out, as I mentioned, emails to everyone who registered to give you a link to the webinar when it's available on demand. So you can definitely come back and check in and see any parts that you missed, or if you aren't in attendance today, you'll be able to check in. So thanks everybody for attending, and you'll hear from us soon. Have a great rest of your day.